friends, and welcome back to another Whispering Wikipedia. Today I am super excited because we are reading about the Venus Flytrap, the plant that snaps shut on its prey, and I think that's super badass and incredibly random. And I've always had an interest in the Venus Flytrap. I remember when I was a kid, I would keep some of those, store-bought Venus Flytraps on my windowsill, and I would try to find flies to feed it, and I was always fascinated in how the plant closed and why it had evolved to do such a thing. I found it extremely interesting, and I believe this might be the first ever Whispering Wikipedia about a plant. And so I am very excited because I don't think I've ever read a plant Wikipedia in full. So thank you for giving me this opportunity to feed my addiction, my Wikipedia addiction, and read a full article. If you have anything you would like me to read about, please tell me in the comments below, because I read every comment and I try to respond to every single comment that I can. I reply to every single comment that YouTube Creator Studio allows me to respond to. Now, let's get started on the article, but please remember that I cannot pronounce scientific names or scientific terminology. And without further ado, let's commence the reading. The Venus Flytrap. The Venus Flytrap, Dinonia muscipula, is a carnivorous plant native to subtropical wetlands on the east coast of the United States in North Carolina and South Carolina. Congratulations to the US for making such a super cool plant. It catches its prey, chiefly insects and arachnids, with a trapping structure formed by the terminal portion of each of the plant's leaves, which is triggered by tiny hairs called trigger hairs or sensitive hairs on their inner surfaces. Interesting. When an insect or spider crawling along the leaves contacts a hair, the trap prepares to close, snapping shut if another contact occurs within approximately 20 seconds of the first strike. I find this fact extremely interesting because, it's a very practical evolutionary thing to make sure that the insect is indeed alive. But I find it crazy because it means that Venus flytraps have within themselves some ability to tell time. I don't mean consciously, of course, but there is some mechanism which allows for them to, be able to tell if the second hair touch has been before or after 20 seconds. And I would love to find out how they do that. Triggers may occur if 1/10 of the insect is within contact. The requirement of redundant triggering in this mechanism serves as a safeguard against wasting energy by trapping objects with no nutritional value, and the plant will only begin digestion after 5 more stimuli to ensure it has caught a live bug worthy of consumption. Wow. Dionaea is a is Aldrophanda vesculosa, and Sundews Drosera, all of which belong to the family Droseraceae. Although widely cultivated for sale, the population of the Venus flytrap has been rapidly declining in its native range. It's not good to hear. The species is currently under Endangered Species Act review by the US Fish and Wildlife Service. Interesting. Maybe one day we will revisit this page and find out the conclusion of the review. Onto the etymology. The plant's common name refers to Venus, the Roman goddess of love. The genus name, Dionia, daughter of Dione, refers to the Greek goddess Aphrodite, while the species name Muscopula is Latin for both mouse trap and fly trap. The Latin word muscipula, mousetrap, is derived from mus, mouse, and decupula, which is trap, while the homonym word muscipula, fly trap, is derived from musca, fly, and decipula, which is trap. Historically, the plant was also known by the slang term dip-it-twitchet, or dippity-twitchet, possibly an oblique reference to the plant's resemblance to human female genitalia. I did not know that. The term is similar to the term tippet-de-witchet, which derives from tippet and witchet, which is an archaic term for vagina. In contrast, the English botanist John Ellis, who gave the plant its scientific name in 1768, wrote that the plant name dippywitchet was an indigenous word from either Cherokee or Catawba. I'm sorry that I most likely slaughtered those two names. The plant name, according to the Handbook of American Indians, derives from the Lenape word dippywitchet, they leaves which wind around or involve. Interesting. Let's go to the discovery. On the 2nd of April, 1759, the North Carolina colonial governor Arthur Dobbs penned the first written description of the plant in a letter to English botanist Peter Collinson. In the letter, he wrote, "We have a kind of catch-fly sensitive which closes upon anything that touches it. It grows in latitude 34 but not in 35. I will try to save the seed here." A year later, Dobbs went into greater detail about the plant in a letter to Collinson dated Brunswick, 24th of January, 1760. The great wonder of the vegetable kingdom is a very curious unknown species of sensitive. It is a dwarf plant. The leaves are like a narrow segment of a sphere, consisting of two parts, like the cap of a spring purse, the concave part outwards, each of which falls back, with indented edges like an iron spring fox trap, upon anything touching the leaves or fall between them. They instantly close like a spring trap, and confine any insect or anything that falls between them. I have given the name of flytrap sensitive Arthropods. This was the first detailed recorded notice of the plant by Europeans. The description was before John Ellis's letter to the London Magazine on the 1st of September 1768, and his letter to Carl Linnaeus on the 23rd of September 1768, in which he described the plant and proposed its English name, Venus's Flytrap, and scientific name, Danuya muscipula. Interesting. The Venus Flytrap is a small plant whose structure can be described as a rosette of 4 to 7 leaves, which arise from a short subterranean stem that is actually a bulb-like object. Each stem reaches a maximum size of about 3 to 10 centimeters, depending on the time of year. Longer leaves with robust traps are usually formed after flowering. Flytraps that have more than 7 leaves are colonies formed by rosettes that have been divided beneath the ground. The leaf blade is divided into 2 regions: a flat, heart-shaped photosynthesis-capable petiole, and a pair of terminal lobes hinged at the mid-rib, forming the trap which is the true leaf. The upper surface of these lobes contains red anthocyanin pigments, and its edges secrete mucilage. The lobes exhibit rapid plant movements, snapping shut when stimulated by prey. The trapping mechanism is tripped when prey contacts one of the 3 hair-like trichomes that are found on the upper surface of each of the lobes. The mechanism is so highly specialized that it can distinguish between living prey and non-living prey stimuli, such as falling raindrops. Wow. 2 trigger hairs must be touched in succession with 20 seconds of each other, or 1 hair touched twice in rapid succession, whereupon the lobes of the trap will snap shut, typically in about 1/10 of a second. The edges of the lobes are fringed by stiff hair-like protrusions or cilia, which mesh together and prevent large prey from escaping. These protrusions and the trigger hairs, also known as sensitive hairs, are likely homologous with the tentacles found in this plant's close relatives, the sundews. Scientists have concluded that the snap trap evolved from a fly paper trap similar to that of Drosera. The holes in the meshwork allow small prey to escape, presumably because the benefit that would be obtained from them would be less than the cost of digesting them. If the prey is too small and escapes, the trap will usually reopen within 12 hours. If the prey moves around in the trap, it tightens and digestion begins more quickly. Speed of closing can vary depending on the amount of humidity, light, size of prey, and general growing conditions. The speed which traps close can be used as an indicator of a plant's general health. Venus flytraps are not as humidity dependent as some other carnivorous plants such as Nepenthes, Cephalotus, and Helianthemora, and some Drosera. The Venus Venus flytrap exhibits variations in petiole shape and length, and whether the leaf lies flat on the ground or extends up at an angle of 40 to 60 degrees. The four major forms are typica, the most common, with the broad decumbent petioles; erecta, with leaves at 45-degree angles; linearis, with narrow petioles and leaves at 45 degrees; and filiformis, with extremely narrow or linear petioles. Except for filiformis, all of these can be stages in leaf production of any plant depending on season. Decumbent in summer versus short versus semi-erect in spring. Or the length of photoperiod, long petioles in spring versus short in summer, and the intensity of light, wide petioles in low light intensity versus narrow in bright light. Now on to habitat and distribution. The Venus flytrap is found in nitrogen and phosphorus poor environments such as bogs and wet savannas. Small in stature and slow growing, the Venus flytrap tolerates fire well and depends on periodic burning to suppress its competition. Fire suppression actually threatens its future in the wild. It survives in wet, sandy, and peaty soils, although it has been successfully transplanted and grown in many locales around the world. It is native only to the coastal bogs of North and South Carolina in the United States, specifically within a 100-kilometer, 60-mile radius of Willington, North Carolina. One such place in North Carolina's Green Swamp. There also appears to be a naturalized population of Venus flytraps in northern Florida, as well as an introduced population in western Washington. The nutritional poverty of the soil is the reason it relies on such elaborate traps. Insect prey provide the nitrogen for protein formation that the soil cannot. They tolerate mild winters, and the Venus flytraps that do not grow through a period of winter dormancy will weaken and die after a period of time. They are full sun plants, usually found only in areas with less than 10% canopy coverage. The microhabitat where it thrives is typically sparse with grasses, herbs, sphagnum, and often bare patches where there aren't enough nutrients for non-coniferous plants to survive, or where fires regularly clear competition and prevent cover from forming. Thus, natural fires are an important part of its habitat, requiring every 3 to 5 years in most places for D. muscipula to thrive. After fire, D. muscipula seeds germinate well in ash and sandy soil, with seedlings growing well in the open post-fire conditions. The seeds germinate immediately without a dormant period. Now, onto the distribution. Dionaea muscipula occurs naturally only along the coastal plain of North and South Carolina in the United States, with all known current sites within 90 kilometers of Wilmington, North Carolina. A 1958 survey of herbaria specimens and old documents found 259 sites where the historical record documented the presence of D. muscipula within 21 counties in North and South Carolina. As of 2019, it was considered extirpated in the North Carolina in the inland counties of Moore, Robinson, and Lenoir, as well as as the South Carolina coastal counties of Charleston and Georgetown. Remaining extant populations exist in North Carolina in Beaufort, Craven, Pamlico, Carteret, Jones, Onslow, Dublin, Pender, New Hanover, Brunswick, Columbus, Bladen, Sampson, Cumberland, and Hoke counties, and in South Carolina in Horry County. Moving on to the population. A large-scale survey in 2019 conducted by the North Carolina Natural Heritage Program counted a total of 163,951 individual Venus flytraps in North Carolina and 4,876 in South Carolina, estimating a total of 302,000 individuals remaining in the wild in its native range. This represents a reduction of more than 93% from a 1979 estimate of approximately 4,500,000 individuals. A 1958 study found 259 confirmed extant or historic sites. As of 2016, there were 71 known sites where the plant could be found in the wild. Of these 71, only 20 were classified as having excellent or good long-term viability. Okay. Now onto carnivory. Most carnivorous plants selectively feed on specific prey. This selection is due to the available prey and the type of trap used by the organism. With the Venus flytrap, prey is limited to beetles, spiders, and other crawling arthropods. The Dionaea diet is 33% ant and 30% spider, 10% beetles, and 10% grasshoppers, with fewer than 5% flying insects. Given that Dionaea evolved from an ancestral form of Drosera, which is a carnivorous plant that use a sticky trap instead of a snap trap, the reason for this evolutionary branching becomes clear. Drosera consumes smaller aerial insects, whereas Dionaea consumes larger terrestrial bugs. Dionaea are able to extract more nutrients from these larger bugs. This gives Dionaea an evolutionary advantage over their ancestral sticky trap form. Now onto mechanism of trapping. The Venus flytrap is one of a very small group of plants capable of rapid movement, such as Mimosa pudica, the telegraph plant, sundews, and bladderworts. The mechanism by which the trap snaps shut involves a complex interaction between elasticity, turgor, and growth. The trap only shuts when there have been 2 stimulations of the trigger hairs. This is to avoid inadvertently triggering of the mechanism by dust and other wind-borne debris. In the open, untripped state, the lobes are convex, which means bent outwards, but in the closed state, the lobes are concave, forming a cavity. It is the rapid flipping of this bistable state that closes the trap. But the mechanism by which this occurs is poorly understood. When the trigger hairs are stimulated, an action potential, mostly involving calcium ions, if you want to see what that is, see calcium in biology, is generated, which propagates across the lobes and stimulates cells in the lobes and in the midrib between them. It is hypothesized that there is a threshold of iron buildup for the Venus Flytrap to react to stimulation. The acid growth theory states that individual cells in the outer layers of the lobes and midrib rapidly move hydrogen ions into their cell walls, lowering the pH and loosening the extracellular components, which allows them to swell rapidly by osmosis, thus elongating and changing the shape of the trap lobe. Alternatively, cells in the inner layers of the lobes and midrib may rapidly secrete other ions, allowing water to follow by osmosis and the cells to collapse. Both of these mechanisms may play a role and have some experimental evidence to support them. Flytraps show a clear example of memory in plants. The plant knows if its hair has been touched and remembers that for a few seconds. The plant then recalls that incident if a second touch happens during that time frame and closes. After closing, the flytrap counts additional stimulations of the trigger hairs to 5 total to start the production of digesting enzymes. And then we move over to digestion. If the prey is unable to escape it, it will continue to stimulate the inner surface of the lobes, and this causes a further growth response that forces the edges of the lobes together, eventually sealing the trap hermetically and forming a stomach in which digestion occurs. Release of the digestive enzymes is controlled by the hormone jasmonic acid, the same hormone that triggers the release of toxins as an anti-herbivore defense mechanism in non-carnivorous plants. Once the digestive glands in the leaf lobes have been activated, digestion is catalyzed by hydrolase enzyme secreted by the glands. Oxidative protein modification is likely to be a pre-digestive mechanism used by Dunia muscipula. Aqueous leaf extracts have been found to contain quinones, such as the naphthoquinone plumbagin, that couples to different NADH-dependent diaphoruses to produce superoxide and hydrogen peroxide upon auto-oxidation. Such oxidative modifications could rupture animal cell membranes. Plumbagin is known to induce apoptosis associated with the regulation of the BCL2 family of proteins. When the Dionia extracts were pre-incubated with diaphoruses and NADH in the presence of serum albumin, SA, subsequent trypic digestion of SA was facilitated. Since the secretory glands of Droseraceae contains proteases and possibly other degradative enzymes, it may be that the presence of oxygen-activating redox cofactors function as extracellular predigestive oxidants to render membrane-bound proteins of the prey, insects, susceptible to proteolytic attacks. Digestion takes place about 10 days, after which the prey is reduced to a husk of chitin. The trap then reopens and is ready for use. On to evolution. Carnivory in plants is a very specialized form of foliar feeding and is an adaptation found in several plants that grow in nutrient-poor soil. Carnivorous traps were naturally selected to allow these organisms to compensate for the nutrient deficiencies of their harsh environments and compensate for the reduced photosynthetic benefit. Phylogenetic studies have shown that carnivory in plants is a is shared with only one other carnivorous plant genus, the Aldrovanda. For most of the 20th century, this relationship was thought to be coincidental, more precisely an example of convergent evolution. Some phylogenetic studies even suggest that the closest living relatives to the Aldrovanda were the sun dews. It was not until 2002 that a molecular evolutionary study by analysing combined nuclear and chloroplast DNA sequences indicated that Dionia and Aldrovanda were closely related, and that the snap-trap mechanism evolved only once in a common ancestor of the two genera. Interesting. A 2009 study presented evidence for the evolution of snap-traps of Dionia and Aldrovanda from a flypaper trap like Drosera regia, based on molecular data. The molecular and physiological data imply that Dionia and Aldrovanda snap-traps evolved from the flypaper traps of a common ancestor with Drosera. Pre-adaptations to the evolution of snap-traps were identified in several species of Drosera, such as rapid leaf and tentacle movement. The model proposes that plant carnivory by snap-trap evolved from the flypaper traps driven by increasing prey size. Bigger prey provides a greater nutritional value, but large insects can easily escape the sticky mucilage of flypaper traps. The evolution of snap traps would therefore prevent escape and kleptoparasitism, which is theft of prey captured by the plant before it can derive benefit of it, and would also permit a more complete digestion. A 2016 study of the expression of genes in the plant's leaves as they captured undigested prey was published in the Journal of Genome Research. The gene activation observed in the leaves of the plant gives support to the hypothesis that the carnivorous mechanisms present in the flytrap are a specially adapted version of mechanisms used by non-carnivorous plants to defend against herbivorous insects. In many non-carnivorous plants, jasmonic acid serves as a signaling molecule for the activation of defense mechanisms, such as the production of hydrolases, which can destroy chitin and other molecular components of insect and microbial pests. In the Venus flytrap, the same molecule has been found to be responsible for the activation of the plant's digestive glands. A few hours after the capture of prey, another set of genes is activated inside the glands, the same set of genes that is active in the roots of other plants, allowing them to absorb nutrients. The use of similar biological pathways in the traps are non-carnivorous, as non-carnivorous plants use for other purposes indicates that somewhere in the evolutionary history the Venus flytrap repurposed these genes to facilitate carnivory. Interesting. On to proposed evolutionary history. Carnivorous plants are generally herbaceous, and their traps result of primary growth. They generally do not form readily fossilizable structures such as thick bark or wood. As a result, there is no fossil evidence of the steps that might link Dionia and Aldrovanda or either genus with their common ancestor, Drosera. Nevertheless, it is possible to infer an evolutionary history based on phylogenetic studies of both genera. Researchers have proposed a series of steps that would ultimately result in the complex snap trap mechanism. Here it is. Larger insects walk over the plant instead of flying to it, and are more likely to break free from sticky glands alone. Therefore, a plant with wider leaves, like Drosera falconeri, must have adapted to move the trap and its stalks in directions that maximizes its chance of capturing and retaining such prey. In this particular case, longitudinally. Once adequately wrapped, escape would be more difficult. Evolutionary pressures then selected for plants with shorter response time, in a manner similar to Drosera brumanani or Drosera glanduliger. The faster the closing, the less reliant on flypaper model the plant would be. As the trap became more and more active, the energy required to wrap the prey increased. Plants that could somehow differentiate between actual insects and random detritus/rain droplets would have an advantage, thus explaining the specialization of inner tentacles into trigger hairs. Ultimately, as the plant relied more on closing around the insect rather than gluing them to the leaf surface, the tentacles, so evident in Drosera, would lose their original function altogether, becoming the teeth and trigger hairs, an example of natural selection utilizing pre-existing structures for new functions. Completing the transition, the plant eventually developed the depressed digestive glands found inside the trap, rather than using the juice in the stalks, further differentiating it from the genus Drosera. Phylogenetic studies using molecular characters place the emergence of carnivory in the ancestors of Dionaea muscipula to 85.6 million years ago, and the development of the snap trap in the ancestors of Dionaea and its sister genus Aldrovanda to approximately 48 million years ago. Now reading about the short section on cultivation. Plants can be propagated by seed, taking around 4 to 5 years to reach maturity. More commonly, they are propagated by clonal division in spring or summer. Venus flytraps can also be propagated in vitro using plant tissue culture. Most Venus flytraps found for sale in nurseries, garden centres, have been produced using this method, as this is the most cost-effective way to propagate them on a larger scale. Regardless of the propagation method used, the plants will live for 20 to 30 years if cultivated in the right conditions. Wow. Mine lasted like a month. And cultivars. Venus flytraps are by far the most commonly recognized and cultivated carnivorous plant, and they are frequently sold as house plants. Various cultivars, cultivated varieties, have come into the market through tissue culture of selected genetic mutations, and these plants are raised in large quantities for commercial markets. Now reading about the conservation. Although widely cultivated for sale as house plant, D. muscipula has suffered a significant decline in its population in the wild. The population in its native range is estimated to have decreased 93% since 1979. Status. The species is under Endangered Species Act review by the US Fish and Wildlife Service. The current review commenced in 2018 after an initial 90-day review found that action may be warranted. A previous review in 1993 resulted in a determination that the plant was a potential candidate without sufficient data on vulnerability. The IUCN Red List classifies the species as vulnerable. The state of North Carolina lists Dionaea muscipula as a species of special concern. Vulnerable. In 2010, Science listed it as an Appendix 2 species, and NatureServe classified it as imperiled G2 in a 2018 review. The US Fish and Wildlife Service have not indicated a timeline to conclude its current review of Dionaea muscipula. The Endangered Species Act specifies a 2-year timeline for a species review. However, the species listing process takes 12.1 years on average. Now on to threats. The Venus flytrap is only found in the wild in a very particular set of conditions, requiring flat land with moist, acidic, nutrient-poor soils that receive full sun and burn frequently in forest fires, and is therefore sensitive to many types of disturbance. A 2011 review identified 5 categories of threats for this species: agriculture, road building, biological resource use, poaching and lumber activities, natural systems modification, draining and fire suppression, and pollution, fertilizer. Habitat loss is a major threat to the species. The human population of the coastal Carolinas is rapidly expanding. For example, Brunswick County, North Carolina, which has the largest numbers of Venus flytrap populations, has seen a 27% increase in its human population from 2010 to 2018. As the population grows, residential and commercial development and road building directly eliminate flytrap habitat, while site preparation that entails ditching and draining can dry out soil in surrounding areas, destroying the viability of the species. Additionally, increased recreational use of natural areas in populated areas directly destroys the plants by crushing or uprooting them. On to fire suppression. It's another threat to the Venus flytrap. In the absence of regular fires, shrubs and trees encroach, outcompeting the species and leading to local extirpations. D. muscipula requires fire every 3 to 5 years and best thrives with annual brush fires. Although flytraps and their seeds are typically killed alongside their competition in fires, seeds from flytraps adjacent to the burnt zone propagate quickly in the ash and full sun conditions that occur post a fire disturbance. Because the mature plants and new seedlings are typically destroyed in the regular fires that are necessary to maintain their habitat, D. muscipula's survival relies upon adequate seed production and dispersal from outside the burnt patches back into the burnt habitat, requiring a critical mass of populations and exposing the success of any one population to meta-population dynamics. These dynamics zone there is no source of seeds post-fire. Furthermore, poaching has been another cause of population decline. Harvesting Venus flytraps on public land became illegal in North Carolina in 1958, and since then a legal cultivation industry has formed, growing tens of thousands of flytraps in commercial greenhouses for sale as household plants. Yet, in 2016, the New York Times reported that a demand for wild plants still exists, which has led to a Venus flytrap crime ring. In 2014, the state of North Carolina made Venus flytrap poaching a felony. Since then, several poachers have been charged, with one man receiving 17 months in prison for poaching 970 Venus flytraps, and another man charged with 73 felony counts in 2019. Poachers may do greater harm to the wild populations than a simple count of individuals taken would indicate, as they may selectively harvest the largest plants at a site which have more flowers and fruit, and therefore generate more seeds than smaller plants. Additionally, this species is particularly vulnerable to catastrophic climate events. Most Venus flytrap sites are only 2 to 4 meters, which is 6.5 to 13 feet above sea level, and are located in a region prone to hurricanes, making storm surges and rising sea levels a long-term threat. In 2005, the Venus flytrap was designated as the state carnivorous plant of North Carolina. And at the very end here, there is, interestingly, a short bit on its use in alternative medicine. Venus flytrap extract is available on the market as a herbal remedy, sometimes as the prime ingredient of a patent medicine named carnivora. According to the American Cancer Society, these products are promoted in alternative medicine as a treatment for a variety of human ailments, including HIV, Crohn's disease, and skin cancer, but available scientific evidence does not support the health claims made for the Venus flytrap extract. Wow. That was super random and interesting, and it was a lot longer than I thought it would be. But that was so interesting. I can't believe how illegal poaching Venus flytraps is. I can't believe someone went to jail for 17 months because he poached 970 Venus flytraps. I find that insane. What a random thing to learn. Anyway, thank you so much for watching the video. I really hope that you liked it. It means a lot to me that you watch. If you have any plant or any animal or anything you would like me to whisper about in my Whispering Wikipedia series, please tell me. I would love to know, and I would love to read it